perception and memory, focusing especially on neural mechanisms. Adam uses innovative cutting-edge methodology and analyses involving fMRI and immersive virtual reality and the combination of the two. He's published work that's theoretically important and rigorous, and I encourage you to check out some of his papers if you haven't already. And I'm so excited for your talk, as are many of us, and I'm going to turn it over to you, Adam.
Thank you, Dan. Thank you, Daniela. Thanks everyone for coming. Just been a real pleasure to be here. Real pleasure to be in Vancouver, get to share my work with you guys, having some fun and interesting conversations already. So yeah, and hopefully some good questions at the end of the talk. So today I'm going to tell you about some work that I did during my postdoc at Dartmouth College, and also some work that I'm continuing on now as an assistant professor at the University of Illinois, Urbana-Champaign, exploring the interaction between memory and perception. So as we navigate through the world, we're dynamically exchanging information about our immediate visual surroundings with our memory of the broader spatial environment that's currently out of our field of view. For example, when you're driving around familiar environment like the University of British Columbia campus, you're using your memory to plan where you're going to go and when you're going to turn your vehicle. And you're actively anticipating what you're going to see when you make that turn. In natural behavior, this dynamic interplay between what we're seeing and our predictions about what we're going to do based on our actions is what allows us to feel immersed in our world. And it's so central to our experience that it goes generally unnoticed. But despite being largely unconscious, this top-down information or influence from our memory on our perception is essential for all kinds of natural behaviors. For example, navigation necessarily depends on that interaction between memory and perception. And that's why I've been using scene processing as my model system of studying these interactions. Because every time we're moving our head and redirecting our gaze, we're actively anticipating what we're going to see, and we're using our memories to inform those decisions. And this was what allows us to move our body through space with ease. But this interaction is not only important for navigation. Our memory allows us to make informed eye movements while we're reading to quicken our comprehension of a word, given its location and with respect to a sentence or a paragraph. Our predictions about our environment allow us to make rapid actions to probable stimuli, like when you're playing the card game Spit. As this pile dwindles, you'll become more likely that your card will come up, and you're going to be able to make rapid actions based on that increasing probability. And it's our feedback from our memory into our perceptual systems that's thought to make it possible for us to perform mental imagery and memory recall, which is just so central to the human experience. So how do these processes manifest? Well, it's possible that this could just be speeding of action selection, and that these kinds of predictive processes aren't actually perceptual in nature. But that's not the case. Elegant cognitive psychology studies have shown us that our memory of our our representations of our fovea and our periphery are primed across eye movements, which allow us or shows dynamically that we're exchanging information between these representations. And given it's important in all kinds of natural behaviors, understanding how memory and perception representations are interacting in the brain is essential. So why don't we know how this works? Well, given our understanding of memory and perceptual systems in the brain, it's not obvious how these representations could be brought together at all. So currently, we see the visual system's neural code as being largely organized around image content. And the importance of content to the visual system's neural code is thought to underpin how this system is organized. So for example, this area of the brain on the lateral surface is necessarily involved in our processing of visual scenes. And we have other areas of the visual system that are selectively involved in processing of other important categories of stimuli like bodies, objects, faces, and words. So in the visual system, content is king. On the other hand, we think of the neural code in memory systems as being more abstract or amodal. So memory systems being the hippocampus and the default network. And like the visual system, our conception of the default mode network's organization reflects of how we think of this neural code as being organized. We generally think of these as being large, undifferentiated swaths of cortex that aren't functionally distinct in the way that we suspect the visual system is. And while these two notions are somewhat reasonable in isolation, without a common principle organizing the neural code between these systems as well as their topography, it's not clear how information could be translated between these systems. However, my work is showing that the distinction between these systems is probably not correct. And in particular, how we think about the memory system's organization and its neural code should be revised. So that's the story I'll tell you about today, and what I address in my work overall, which is what mechanisms allow perceptual and mnemonic representations to interact in the brain. In addressing this question, I look at how brain systems topography and connectivity facilitate their interaction. I ask what memory and perceptual systems' unique and shared contributions to cognition are. And I ask what coding principles facilitate the interaction between these representations. So today I'm going to focus on these questions in the domain of scenes. First, talking about the brain's functional organization and how that's facilitating perception and memory interactions. Having described this functional organization and the differentiation between perceptual and mnemonic areas, I'll then talk about what unique information they represent and process. And I'll end by talking about some recent work where I've been examining the neural coding principles that enable these regions, these types of information to interact. So let's get started with topography. So as I said in the beginning, we have a robust understanding of how the visual system is organized, and scene perception is one component of that. So in particular, we think about the brain as having three scene-selective visual areas, one on each of the cortical surfaces. So we have the parahippocampal place area on the ventral surface, the occipital place area, or OPA, on the lateral surface, and the medial place area, or MPA, on the medial surface. Sometimes this is referred to in the literature as the retrosplenial complex. So these regions respond more when viewing images of scenes compared to other types of information, visual images, and damage to these regions causes scene-specific deficits and agnosias. So that's how we know that they're causally involved in scene perception, and I'll refer to these regions collectively as the scene perception areas. So we also know that the hippocampus and the entorhinal cortex are critical for spatial memories and representations, and they actually contain, as you probably know, specialized cells for coding this information, namely place cells and grid cells. However, we know considerably less about how information from the hippocampus and entorhinal cortex is being fed back into the scene perception system in order to facilitate visually guided navigation and processes like that. So how might these representations be brought together? The dominant view is that the integration of memory and perceptual information is happening within shared neural substrates. So the evidence for this comes primarily from studies of mental imagery, which is you can think of as kind of a type of explicit memory recall. And a number of fMRI studies have shown that performing mental imagery and memory recall of visual information like scenes tends to activate the same areas that are processing those information visually, including low-level areas like primary visual cortex, all the way up high level areas like perceptual OPA and PPA. And so this theory suggests that the integration should be happening within this shared region. On the other hand, more recent studies, primarily using resting state functional connectivity analyses, have pointed to the possibility that the anterior aspects of these scene perception areas are more connected among each other and to the hippocampus compared to the posterior aspects of these regions, suggesting that there might be a perceptual to memory-oriented transition within the scene perception system. So there might be a distinction between these. However, this hadn't really been examined within empirical studies outside of resting state functional connectivity. So how do we reconcile these hypotheses? Well, the approach that I took to answering this question was to perform very detail-oriented, individualized analyses of scene perception and memory within individual participants to enable a fine-grained comparison between these processes. So for this study, we performed two different functional localizer tasks. The first localizer is a very vanilla, traditional scene perception localizer. And this is very common in visual neuroscience to localize these scene perception areas among other category selective areas. And in our case, we showed participants blocks of rapidly changing scene images and compared bold activity during those scene blocks compared with other periods of time where the participant was looking at images of faces. The more innovative aspect of this study was what we did to localize the memory responsive areas for scenes, or for what we're calling places. So for this place memory localizer, we had participants provide for us outside the scanner a list of 36 personally familiar people and places that they could visualize richly. So these were people and places that they knew, that they had been to and they interact with in their lives. And then in the scanner, we showed them those same people and places, the names of them, for 10 seconds and had them do a really vivid visual imagery task during that time. So the specific instructions for the participant is that if they were seeing a scene word, they would imagine that that place had materialized all around them, that they should be directing their attention smoothly around that place as though they were looking around it while standing in one location. They shouldn't imagine serially the objects being presented, but instead focus on being in there in an immersive way. For people stimuli, we told our participants to imagine that person's face had materialized in front of them at a comfortable distance, that the person should be turning their head, looking up and down, taking different perspectives and changing emotions. But that it should be very visual in nature. So I think that the way that we gave these specific instructions is very important to the results that we found. And so I'm very happy to talk about those findings that lead me to believe that in questions if people are interested. But with these tasks in hand, we could then run these tasks as an individual participant and look at an individual subject level whether these tasks are activating the same brain waves. So I'll show you a couple example subjects here because these are the data that I actually was having my hands on to perform these analyses. So this is our first participant. This is their ventral surface. You can see the parahippocampal place area in hot colors and the fusiform face area as a control region in cool colors. This is OPA on the lateral surface and then MPA on the medial surface. So again, these are where scene selective perceptual activity is occurring. Now going to show you where the memory activity was, and I'll focus first just at OPA on the lateral surface. So this is OPA. It's going to remain outlined in white when I show you the contrast between our place versus people memory task. So again, this is perception. And here's what we see during our memory task. You can see there's a dramatic anterior shift of our memory selective activity relative to that perceptual activity on the lateral surface. And there's really only a very small region of overlap between perceptual OPA and this mnemonic region that we're calling the lateral place memory area. So that's what happened on the lateral surface. I'll show you all of the surfaces now. Again, the perceptual areas are going to be outlined with white. Here's that memory selective activation. And you can see a dramatic anterior shift of memory compared to perception on each of the cortical surfaces, although the precise nature of that shift does depend on the surface. So in the medial surface, we have this general expansion of what you see in the medial place area, but it tends to expand anterior and ventrally down the parietal occipital sulcus towards retrosplenial cortex proper. And on the ventral surface, we have an anterior extension of PPA, although there's a large overlapping zone. The selective activity tends to spill out anteriorly to where that perceptual activity ends. And on the ventral surface, or if you consider the face activation here, you can see that there isn't such a dramatic shift of the face selective activity compared in memory compared to perception. There's a slight lateral shift, but in general, it's mostly confined within, or it is entirely confined within that larger face selective perceptual patch. So that's one subject. Here's a second subject. Again, you can see their scene selective activity in hot colors and their face selective perceptual activity in cool colors. And here's their memory related activity. Again, on the lateral surface, you can see that really dramatic anterior and dorsal shift of perception of memory compared to perception. But again, on the ventral surface and the medial surface, you see the large anterior shift in the activation compared to what you see during perception. And for faces here, you see the activity is entirely confined to within a perceptual region. So no real shift to speak of for face memory compared to perception. So I would love to go through all of the participants. It does give me great joy. Instead of doing that though, I'll show you the data collapsed in the format of these rose plots. So the way you read these rose plots is that the center of mass of perception is the center of the plot. The stem of the rose represents the shift with respect to the center of mass of that memory related cluster. So we're basically trying to visually depict what you were doing with your eyes in that previous slide. And the distance is going to be millimeters along the cortical surface. So I'll start again with the lateral surface here. You can see that every single participant that we scanned in this study showed an anterior and dorsal shift of the memory cluster compared to the perceptual cluster. And the average shift is about 2 centimeters. So it's a pretty significant shift. Here's the medial surface. You can see all of the subjects are showing an anterior shift, and most of them are showing that anterior and ventral shift in activity. So going down the parietal occipital sulcus. Here's the ventral surface. It's pretty much a veridical anterior shift of about a centimeter and a half on average across our participants. So again, a huge anterior shift there. And we don't see that same large anterior shift for faces. If anything, there's evidence for a slightly lateral shift like you saw in that one participant, but it is about an order of magnitude smaller than what you see for the shift that you see for scenes. So it suggests that this might be a scene specific effect, and that's something that we've been very interested in following up. So for the imaging aficionados in the room, you'll note that the face selective patch on the ventral surface is like quite lateral compared to the scene selective patch. So it is impacted by the dropout artifact in ventrotemporal cortex. So we actually reran the study with multi-echo fMRI to try to mitigate those dropout artifacts. We were able to replicate the same findings, an anterior shift for scenes and no significant anterior shift for faces once we ran the same study with very short T multi-echo fMRI. So we run this localizer in a number of participants now. We've actually generated a probabilistic atlas of the locations of these regions from all of our subjects that we've run. This is just the lateral surface depiction of those findings. So this is a probabilistic cluster of the overlap of those significant regions of interest. Of where those regions of interest are. This is the scene selective patch. This circle is the peak of probability, and you can see here's the memory area region of interest and its respective peak in probability. So you can see that pretty significant anterior shift across all of our participants. Yes. Yeah, absolutely. Yeah. Yeah. So you mean the T statistic? So it's a contrast between our scene selective, our place memory recall compared to people memory recall to contrast those. For perception, it was scene selective perception, so scene images versus face images. We've done a number of different stimulus classes, so objects, including objects in the contrast, for example. You still see that significant anterior shift. Another bit that might be implicit in your question is, what about thresholding related issues? So is this simply an artifact of having your T statistic thresholded at a certain level? These don't seem to be T statistic or thresholding related features. So we have in the manuscript all of our subjects unthresholded maps as one way of looking at that. Another way of looking at that is looking at what the steepness of the fall off is of the memory selective activity. And what we find is that steepness falls off in the posterior portion of that cluster at about the same distance as our smoothing kernel. So that's another feature that gives us some confidence in the fMRI data that it's not about thresholding related issues. It's really a topographic property of the regions that are being have this evoked selectively. Does that answer your question? Yeah. So you mean is the percent signal change comparable between our memory and our perception task? Is that what you're asking? The betas of scenes versus faces and their respective memory conditions. But we do run those conditions, those tasks separately. And that's by design, because actually in our first study, we were concerned that there might be a general lower level of activation for our memory task compared to the perception task. In fact, that's not what we see when we run them in the same scan session. That memory and perceptual activity, the magnitude of the activation does seem to have this shift. I could go into more detail about why we had expected, but hopefully that answers your question. Yeah, contrasted betas. That's correct. Yeah. Well, the memory and perception, the scene versus faces within each task were run at the same time, but across tasks they were run at different times. So in an imaging run, it would be a memory run, and we would have scene versus faces intermixed. Does that make sense? Yeah. Okay. So just one quick feature that we can get now that we've generated these probabilistic clusters is looking at other features of organization of large-scale cortical organization and thinking about how our clusters map onto those. So one very popular organizing principle lately is the idea of a large-scale cortical gradient. This is popularized by Margulies and colleagues. For those who might not be familiar with it, the general principle here is that the idea that across the cortical surface, we have this smooth transition from unimodal sensory or motor areas into these higher level transmodal regions, like associated with the default mode network, for example. So we can look at where our clusters fall with respect to those gradients. So I'm going to show you that. I'm going to leave the memory cluster outlined in red and the perceptual cluster outlined in white here. So here's this gradient, this kind of abstractness gradient, if you will, from Margulies and colleagues. Here's our perceptual cluster, here's our memory cluster, And what you can see is that our perceptual cluster is falling largely within the unimodal portion of that gradient. And if I plot gradient value here and our frequency of nodes or frequency of surface vertices compared to that gradient value, you can see consistent with, the data is consistent with that claim. So on the other hand, our memory responsive region seems to be centered on this inflection point between unimodal transitioning into transmodal cortex. And that is in fact what we see when we compare the gradient values there. It's a significant shift and really spanning the full spectrum of the gradient from unimodal to transmodal cortical areas. So we think is, again, it's quite a bit an exciting organizing feature and an interesting bridging topography of this memory responsive patch. So having defined these regions, our next question is whether or not these regions are actually functionally distinct. And the way we've decided, or the way I decided to take a look at that is by asking whether they constitute a distinct functional network. So what we would expect to find if these networks are distinct is that the memory responsive regions and the scene responsive, perception responsive regions should form separable networks with the perceptual regions having activity that's more correlated among each other and the place memory regions having activity that's more correlated among each other. And based on our supposition of their functional properties, the scene responsive cluster should be more connected with visual cortex, and the memory responsive cluster should be more connected with the hippocampus. So we tested this by looking at the co-fluctuation of these regions while our participants watched this naturalistic movie. So the video, the movie that they watched was a series of videos that I took from YouTube, which were architectural tours, real estate advertisements, college campus videos, and generally videos that were meant to convey a strong sense of space. And I chose movie watching rather than resting state fMRI because during resting state, people are often doing some kind of like mental imagery or self-projection into other places that they might be, or thinking about people that they're going to interact with, which could drive a distinction between these areas artificially. So by having them attending to a visual scene, we would expect that it would actually be harder to find any distinction between the perceptual and memory areas. So here you've got the within network and between network correlations of their activity on the x-axis. On the y-axis here, you have the correlation values. The within network correlation between these two networks was comparable. And we found that the within network correlation was comparable and quite strong, and the between network correlation was significantly reduced, suggesting that they are forming these distinct functional networks during this naturalistic movie watching class. And we found that they did form these dissociable connections with primary visual cortex and hippocampus that reinforce our idea that the scene perception areas should be more perceptual in nature given their correlation with V1, and that the place memory areas are more mnemonic given their correlation with the hippocampus. So this suggests dissociable roles for these networks, and actually based on their topography and correlational connectivity, suggests that the place memory areas are actually in a position to bridge those perceptual and mnemonic representations. So to conclude this section of the talk, what I've shown you is that there's a network of brain regions that lie immediately anterior to the scene perception regions that are preferentially supporting our perception and memory of familiar scenes. So having described these regions, the next important question is why there should be this distinction. Why should it be the case that there is this dissociable topographic representation of topographic processing of perceptual and mnemonic areas? And the obvious prediction is that the memory areas should be involved in memory, right? But what is the specific nature of that memory? Memory-related information. So the specific kind of memory that related information that we hypothesize these regions should be processing is visuospatial context, that knowledge of the visuospatial information that's outside of your current field of view. And we couldn't test that in our previous data because we were using personally familiar scenes. So these are scenes that you have all of this embedded semantic and episodic details that we couldn't disambiguate from our within our memory task. So in order to focus our participants on just the visuospatial context, we had them train in virtual reality to learn a set of environments. And crucially, what we could manipulate with VR is the amount of visuospatial context that was associated with those visual environments. So the environments were split into 3 different conditions: an image, a panorama, and a street condition. And I'll show you video examples of each of these trials that the participants experienced during training. So in the image condition, the participant donned the headset. They saw the name of the place that they were going to be immersed in, and they would see a sliver of that image. So it was just the inner 45 degrees of that image. They could still turn their heads and move their bodies to look around them, but they couldn't see anything except what was in that narrow aperture. In the panorama condition, the participant again sees the name of the place. They press the trigger to start, and then they can look all around them. And 270 degrees of their environment is all open for them to visually explore. If they turn around and look directly behind them, that information, that last 90 degrees, was occluded by a gray screen. But everything else they could explore naturally with their eyes and bodies. In the street condition, finally, they saw the name of the place, they pressed the trigger to start, and they're immersed in the 360-degree panorama. They can turn all the way around, and when they do, they see this arrow on the ground that indicates that they can take 3 virtual steps forward by pressing a trigger on a controller. And once they get to that 3rd photosphere, they could turn and look around a corner and see visual information that they couldn't see from their initial standing position. So in that way, it's kind of like Panorama Plus. They can reveal additional visuospatial information that they wouldn't have been able to see. So we trained participants until they could recall these scenes accurately, and then let them go home to sleep. And on the 2nd day, and actually a 3rd day, had them do a recall and a recognition task in the MRI scan. So in the recall task, it looked almost identical to what we had done previously, except we showed them this dynamic mass after the cue, and we cued them to perform recall with these 4 circles that appeared on the screen. They would do the recall for that whole 10-second period, and then they would see a fixation cross. And in our instructions, instead of asking the participants to imagine looking all the way around them, we told them to just imagine your initial viewing position and hold that mental image in mind as well as you can. So trying to get them to focus only on one field of view and not what was out of context. And what we predicted is that a brain region that is involved in processing this visuospatial context should have greater activity across the conditions, with panorama being more than image and street being more than image as well. So this was our prediction. What did we find in our tasks? So here's the activity from our perception and memory areas from this experiment. Here's our image, panorama, and street for our perception activity in pink and our memory-related activity in maroon. And on the y-axis here, you have activity during recall. This is versus baseline, fixation baseline for reference. So what we found is that this is just data from the lateral surface. Also, we found comparable results on the other cortical surfaces, and I'm happy to show those data at the end of the talk if there's interest. So what we found is that in the memory area, we see that parametrically increasing activity across our image and panorama and street conditions, and activity above baseline during our memory task. suggesting that this area is involved in supporting memory and also processing visuospatial context. And critically, versus our fixation baseline, we see that there is no activity within this memory perceptually responsive region, and also no modulation by visuospatial context within this region. So that was memory recall. We also had them perform a perceptual recognition task. And in the recognition task, we had them just perceive the inner 45 degrees of their initial standing position, and then they had to indicate on their indicate to us whether they had studied that image during VR or not. And we had several perceptually matched foil images also intermixed. And this task is critical because, of course, in this memory task, we asked them to just hold one image in mind, but of course, they could be doing whatever they want. But here, we're only showing them that inner 45 degrees, so we know exactly what they're experiencing. They are making that judgment that is on the image in the 45 degrees. So any activity, we suspect that any activity that's active here that shows the spatial context modulation is because the region is implicitly processing the information outside of view. So again, perceptual area, memory area, activation on that y-axis. Yeah. No, this is versus fixation baseline also. We didn't have the same number of foil trials, so we thought that the fixation baseline was a better comparison for these conditions. This is just the beta values versus our fixation baseline. But the critical comparison, of course, is not the activity relative to baseline, but actually across these conditions, right? That's what we really want to see. So this is just a way of visualizing those data across the conditions. Does that make sense? Okay. So what we find is that in our in our recognition task, our perceptual area is active above baseline, our fixation baseline, which is important because it is a perceptual area. We should expect that it will be more active when you are perceiving scenes compared to looking at a fixation cross. But importantly, there's no significant modulation across these spatial context conditions. In the memory area, we do see a strong modulation by these spatial context conditions, again, with image less than panorama, less than street. And there is a significant difference between these regions. So together, these data suggest that the place memory areas are in fact processing the visuospatial information that's outside of view, both during memory recall and also during perception. So that is the conclusion of the second portion of the talk. Last, I want to talk about how we're going to bring perception and memory information together, right? Because fundamentally, we know that these regions are interacting. They might be supporting different processes, but we think that that memory-related information should be getting into the perceptual system in order for us to do predictive vision-like tasks, for example. So all the data that I've shown has suggested that the memory and perception areas are working together to support perception of familiar scenes. But what mechanism allows this information to be shared between these brain areas? This is generally a puzzle, again, because we think that the perceptual and memory areas should be using distinct neural codes, and that the memory areas in particular should not be using a visual code to process their information. So visual cortex encodes information retinotopically, such that neurons respond when specific portions of the retina are stimulated. And using fMRI, we can measure these receptive fields at the level of voxels, which we call population receptive fields. And throughout much of visual cortex, retinotopically responsive neurons and voxels are organized as maps of the retina recapitulated on the cortical surface. So here I'm showing the known retinotopic maps in dotted lines. And what you can see is that these maps overlap with our perceptual, scene-selected perceptual area, consistent with this region using a visually grounded neural code. On the other hand, the memory area is largely anterior to these maps. So we wouldn't expect the memory area to be organized retinotopically, and we wouldn't expect it to be implementing a retinotopic code. On the other hand, the memory area tends to fall on the edge of the default network. And the default network is thought to use that abstract or amodal coding scheme. So this is our puzzle. How can we get information to be translated between these memory and perceptual areas if they're not using similar neural codes? So prior work has begun to suggest that retinotopic coding might persist in memory areas and in the hippocampus. And so here we sought out to test that notion, whether the place memory and scene perception areas are both exhibiting a retinotopic code, as this literature has begun to suggest, and further, whether that retinotopic code is actually going to be structuring the interactions between these regions. So to address this question, we recruited 17 participants for a study in which we did population receptive field modeling. So for those who might not be familiar, this is how population receptive field modeling works. We're going to take, for example, a voxel and visual cortex. That voxel's receptive field I've shown here with this red circle. And we're going to move this bar stimulus across the visual field and record fMRI activity. So right now, this bar is not in that voxel's receptive field, so a population receptive field, so we don't see any change in that voxel's response. When the bar enters that voxel's receptive field, it evokes a positive gold response. We sweep the bar in different angles and directions, and every time that bar enters the voxel's population receptive field, it evokes a gold response. Importantly, the participant is maintaining fixation the whole time. We have them doing an attention task, which is, in our case, a color changing dot that they need to detect. So in total, we do 8 different bar sweeps with 4 different angles in 2 different directions. We then use a computational model to predict what a voxel's response would be given a particular center position and size. And we fit that to each voxel's data by modulating an amplitude parameter, which is essentially how much that voxel responds to a visual stimulus in percent signal change. So in our paradigm, we don't show that funny pattern inside the bars. Instead, because we're dealing with scene-selected perceptual areas, we actually show images of scenes that are rapidly changing inside this bar aperture. We fit our PRF model to all voxels in the brain. And importantly, in this study, we're choosing a voxel-wise R-squared value of 8% to define voxels that we are considering retinotopic. So I did find that PRFs are in our memory areas, which is exciting and new. This is not what is expected given the dogma of how neural coding should work outside of visual cortex. But when we looked at the visual responses of those voxels in our memory area, that's where we found something that we considered quite remarkable. So as you would expect, we found that population receptive fields in our scene perceptual area OPA have a positive bold response when their population receptive field is stimulated. So if we just take a cartoon example here, we have a tree. This tree is not in this PRF's, this OPA PRF's receptive field, and so there's no signal chain. If we make an eye movement, for example, and move that tree into the receptive fields, it evokes a positive bold response. The receptive fields in our memory areas, like LPMA, had a very different response property, actually diametrically opposed. So when their receptive fields are stimulated, we see a negative bold response compared to that voxel's estimated baseline response. This is a spatially specific negative bold response. I refer to these voxels with negative bold responses as negative PRFs for the rest of the talk. So these response properties are weird to think about in the abstract. So here's 2 different example voxels and their time series from this task. So this is the positive PRF from OPA. you can see that this voxel baseline response, which again is estimated when that voxel PRF is not being stimulated, and whenever it is stimulated, it has a positive fold response relative to that estimated baseline. And here's our negative PRF. Again, compared to periods of time when that voxel PRF is not being stimulated, it evokes systematic negative fold responses when the bar traverses that population receptive field. And importantly, these receptive fields are very similar in the visual field. And so these responses are essentially anti-correlated based on the location of these receptive fields. So just how prevalent are these negative PRFs in cortex or in our scene perception and memory areas? So again, I'm just showing you the data from the lateral surface here, but I'm happy to show you the other surfaces of the brain during Q&A if there's interest. So this is OPA, this is LPMA. On the y-axis here, you have the proportion of percent of negative PRFs that we find in those regions. Our OPA PRFs are primarily positive. So across our participants, only about 3% of the population receptive fields in OPA are negative. On the other hand, we find that LPMA has significantly more negative PRFs. And there's about 60% negative PRFs on average, with some subjects having as many as 100% negative PRFs in this region. So negative PRFs are much more prevalent in the negative PRFs are much more prevalent in this memory area compared to the perceptual area. So what this suggests is that the memory areas are implementing a retinotopic code. And the critical question is whether this retinotopic code is playing a functional role in structuring these regions' interactions, right? So if this were true, we should observe two features. First, we should see that the scene perception and place memory areas that are paired with each other should be sharing visual information. And second, we should see that this same retinotopic code of these paired regions should be structuring these regions' interaction during memory recall. And I'll unpack both of these in turn. So first, perception. The scene perception areas, OPA and PPA on the lateral and ventral surfaces, have differential representations of the visual field. They have biased visual field representations, such that OPA tends to overrepresent the lower visual field, and PPA tends to overrepresent the upper visual field. Here I'm quantifying those visual field representations where negative values are greater representation of the lower visual field, or a greater preference for stimuli that are appearing in the lower visual field. And you can see this bias clearly with PPA preferring the upper and OPA preferring the lower visual field. The question is, do the memory areas share these biases, which is what we would expect if they're inheriting the information from vision in a bottom-up way. And that's what we find. The negative PRFs in our memory areas recapitulate the biases of their paired perceptual areas. And this is consistent with a close interaction during of the negative PRFs in the memory areas and the positive PRFs in the perception areas during perception. So we wanted to, I suppose, we wanted to find out what happens when memory is in the driver's seat. Specifically, what's happening when participants engage in top-down memory recall. So just to return to our example of the palm tree, this is what we see during perception is positive activity in the perception areas and negative activity in the memory areas, negative bold responses in the memory areas. What should we expect during memory? Well, this pattern should be flipped. We should see more activation in the memory areas, and our scene selective positive PRFs should be relatively suppressed. And crucially, if retinotopic coding is structuring that interaction, that negative correlation of their activity should be stronger for population receptive fields that are representing similar portions of visual space. So we tested this by matching population receptive fields between these areas based on their visual response preferences. and looking at how those matched visual response preferences correlate with the activity that these regions have during our memory task. To make this concrete, we take each OPA PRF, find its best matched negative PRF and LPMA, and then we randomly pair it with other negative PRFs that are not matched for visual representation. And we do this matching and randomization procedure 1,000 times. What we expect to find is a negative correlation on a trial-by-trial basis between the negative PRFs in the memory area and the positive PRFs in the perception area, such that on trials where the memory area activity is high, perception area activity is low. Negative when memory activity is low, perception area activity should be high. And this pattern should be consistent across all trials in the participant. And this negative correlation should be modulated by the voxel's matched visual field preferences. So this is the data from all of the trials from this memory task in our participants. You can see the negative correlation is present, consistent with that opponent interaction, that anti-correlation we expect. We can summarize this for each individual by looking at their unique correlation value. And this is for matched PRFs. Again, you can see it's negative. What happens in our randomly matched PRFs? That correlation goes away. So there's a strong negative correlation for PRFs when they are matched for their visual field preferences, suggesting that this retinotopic code between these areas is structuring their interactions during this task. So yes, remarkably, we find evidence that this retinotopic code is playing a role in structuring the memory and perceptual areas interactions during recall. So this is just a summary of those findings. We do find retinotopic coding in the memory areas. Retinotopic code is structuring their interactions. One question you might have is just how general is this retinotopic scaffold? So we've been exploring that. What you're seeing here are the memory and perceptual areas on the lateral surface. And of course, they're only a small fraction of the default mode network's topography. So I'll show you next is a map of what the PRF amplitude looks like across cortex, where hot colors are positive PRFs and cool colors are negative PRFs. So any voxel that's shown here survives our critical R-squared threshold. And what you can see is that negative PRFs tend to be clustered within the default network, as well as in our memory areas, compared to positive PRFs that tend to be in our visually responsive regions. And so we've been looking at just how widespread this interaction is between our negative and positive PRFs. I'll just show you one replication that we have in a resting state fMRI data set, where we looked at visually responsive dorsal attention network PRFs being matched with PRFs within the default network. So this is looking at across the whole network, is retinotopic coding structuring their interactions? Again, using that same visual field matching procedure I explained previously. So on a voxel-wise basis, we can assess whether there's a stronger correlation in resting state when there are no visual demands between our matched PRFs and our anti-match or our randomly matched PRFs across these networks. And we do find that this is the case during rest. Anti-matched PRFs are negatively correlated, but the matched PRFs are significantly more anti-correlated, suggesting that the interaction between these networks is structured by this retinotopic code. And we also find evidence that this is true within visually responsive voxels that we can identify in the hippocampus, such that well-matched PRF, positive and negative PRFs in the hippocampus are also more negatively correlated than PRFs that are randomly matched. So I think this is a really exciting direction to pursue, thinking about how a visuospatial scaffold might be structuring the interaction, not just between visually responsive regions, but really a mechanism for interactions across the whole brain at a voxel-specific level. The hierarchical kind of organizing principle that is underneath what we are seeing when we look at whole networks interacting. So that's all I've got for today. Just to conclude, I've described a set of brain areas that are involved in perception and memory of familiar places. The memory responsive areas are processing the visuospatial context that's outside of the current field of view. And these regions seem to be interacting in a retinotopic, using a retinotopic coding scheme that's organizing their interactions during memory recall, scene perception, and also spontaneous activity during rest. So this is emblematic of the research program that I'm pursuing at the University of Illinois. And these are my wonderful colleagues from the Robertson Lab, Caroline Robertson, who is my postdoc supervisor, who are instrumental in this work. Peter Angeli has been working very closely with me on the resting state data. Ed Stilson has been a longtime collaborator and friend, doing a lot of this work on memory and population receptive field modeling. And that's all I have. Thank you.
Okay, we have lots of time for questions. Debbie, I'm going to bring you a microphone.
That was great, really interesting. So I'm wondering, because this part of the brain shows some retinotopy, when people are recalling the scenes, do they recall them in the center of their visual field?
Yeah.
Like, could have maybe seen them up here?
Yeah.
And that's why you see the shift?
That's a really great question, and it's something that Ed and I in particular are thinking a lot about. So Ed's running some behavioral studies right now looking at how people do position-specific memory. Just like a quick summary of what he's finding is that people do tend, if you given unlimited degrees of freedom, they do tend to put their visual imagery in the center of the visual field. Scenes do tend to have a wider expanse in the mind's eye compared to faces, which tend to be more foveal. All of this matches our expectation. One thing that we did try in this experiment, because we were interested in whether this is position specific, is actually having people do the position specific imagery with visual scenes. And there we got some kind of counterintuitive behavioral reports from our participants that made us feel like that wasn't going to be a productive way to go. And this is that the participants, when they were doing scene imagery, just couldn't do it. Yeah. They couldn't deploy a scene image into a visual field quadrant. There were two kind of strategies that the person would use. The first strategy was they would kind of populate a full field visual scene and then direct their attention as though they were going to make an eye movement. So it was like highlighting a particular area. And the other strategy that people would use was populating a full field image and then suppressing everything else. So trying to basically like dim the brightness of the mental image that was not in the area that they were supposed to be attending. So those were the two strategies. You could argue that they're just two sides of the same coin. But because we were interested in visual scenes and not faces or objects, we went away from the strategy of having people do position-specific imagery. We do think that people can do position-specific imagery for objects and faces. There's some good data from Sarah Fabula is one of the lead authors on this, and also Jesse Breedlove working with Thomas Nazalaris has a paper on this, where people are deploying position-specific imagery of objects. And so it's definitely possible, but I just think that scenes are a little different than what people do with with objects and faces. And that's why we didn't address it with this study. But I do think that this could be a mechanism for doing the position-specific imagery for these other visual categories. And I'm very interested in pursuing that. Yeah.
Since I'm holding the microphone, I'm going to ask the next question. That was a fantastic talk, super interesting. I have a question about your first and to some extent your second study. I want to ask you about like whether you think your findings are generalized to different kinds of memory. So for example, your first task of imagining your mother, different orientations, I think of that a little bit more like semantic memory. And then I also think that places a lot of constructive demands on the participant. So I guess I have two questions. One is about whether you think some of these findings would extend to other kinds of memory. And then what you think of kind of this idea that anterior regions are also kind of implicated in more constructive processes, certainly within imagination. So yeah, if you can address both of those.
Yeah, that's a great. I feel like there are two kinds of questions in there. Both are very good. I'll take them what I think are in turn. So one question is about different types of memory, being class domains of memory rather than visual categories. So like you could be thinking about things like semantic memory or emotional memory or these other classes of things. I don't have a strong explanation or hypothesis there. We've had people do imagery tasks of uh, audition compared to like auditory imagery compared to auditory perception, and there doesn't seem like there's this systematic anterior shift. There's some differences, but it's not this same categorical like kind of large scale activity cluster shifting that is systematically anterior and adjacent that we see for scenes. And that leads me to believe that to some extent scenes are special in having this overlapping, like anterior and overlapping zone that is just different in kind from what you have in other domains, probably more related to the kind of demand that scenes and navigation in particular, where you're immersed in the thing, make sense of it in space. So you need to kind of have this dual representation of the current visual field and the information that's kind of it is embedded within. So that's one question. I'm trying to remember now the other half of your question.
Yeah, my other question was about just constructive demand.
Yeah, constructive demands. So that's a great question. It's definitely the case that the scene construction task that we have is visually demanding. I can take this in a couple different directions. One is a domain specific thing, and the other is like about scene construction in particular. So I'll go scene construction in particular, and we can back up and talk about the other thing. So scene construction in particular, the nature of the visual demand that is implied seems to shift the anteriorness of this activity. So in the case where I'm working right now on some data with Lauren DeNicola, who does some more of this scene construction work. So in their task, they're doing this temporal projection into past or future that has a visual demand, but it's not explicitly a visual task. And when they have to do this temporal projection, it seems like the activity is even more anteriorly shifted, but the amount of that anterior shift seems to depend on how visual in nature the task, like at a trial by trial basis, what she has shown is that the magnitude might change depending on how visual the participant is being compared to how temporal and episodic projection they're being. So whether it's sensory grounded. And our task, because we're having them do something that's very sensory grounded, we get this overlapping thing, which suggests that there's this gradient from vision to more abstract representation in the constructive nature. Hopefully that answers your question.
It totally answers my question. It's super clear. Thank you so much.
Yeah.
Other questions?
So an ongoing debate in imagery perception in terms of how they interact is whether they can both simultaneously operate. And so when perception is online, maybe imagery has to take a back seat. And then when imagery is engaged, then perception sort of has to take a back seat, right? And so if I ask you to imagine how many windows were in your childhood home, you like avert your gaze because you would need to disengage from visual input in order to construct the image. And so I'm curious whether your data, whether you would sort of, this work would suggest that kind of dissociation where it's like, yes, they actually would, or they can't operate together simultaneously. Which then, in which case, I would be interested in sort of going back to how you opened your talk, which is how, when we're navigating the world, it seems like we are still recruiting some bit of memory components. And so in online navigation or perception, what then would the role of these place memory areas be, you think?
Yeah. I think it's a great set of questions. So with respect to the imagery debate and the like kind of suppressive, the opponent or competitive interaction between them, perception. I think these data are very consistent with that, where we have memory-related activity driving up the memory areas and down. We do seem to find evidence for that competitive interaction. I think that this actually gives us a very specific prediction, which is that beyond a global competition, that if we deploy mnemonic, imagery spatially, that we should see selective changes in our memory recall or encoding capacity, depending on where we've deployed our attention during encoding, or deployed our attention or imagery during the encoding phase. So we should have this not only a general competitiveness, but actually a position-specific competitiveness, and that's a behavioral question I'm very excited to address. With respect to what are these memory responsive regions doing, and particularly during navigation. So I have never been a person who really cared deeply about imagery. And so I've kind of like wandered blindly into the imagery space because I really wanted to know what outside of perception, like what is the visual system doing outside of the context of immediate perception. And I have always been grounding in what is the visual system trying to do. And what the visual system in this case is trying to do is to, in my mind, is to plan where you're going to direct your gaze next and what things should be falling into your fovea based on where you are directing your attention. So I think that the mental imagery system is like this weird ability that humans have that bootstraps on top of this system that's really just doing eye movement prediction. And that we have this conscious or phenomenological ability to like do that over an extended duration of time. So my expectation is that during navigation, that what the memory areas are doing are kind of selectively engaging in depressive or competitive processes, depending on what you're anticipating putting into your current field of view, based on what you're currently attending to. Yeah, so happy to take more.
Yeah, it would be really cool if you saw that kind of anticipatory where right, like you see something activated within perception areas, but then what's in the place memory areas in online navigation is actually a step ahead of what might be going. Like that would be really, and you could do a one-to-one mapping if you're doing it in navigation. So that could be really cool.
think that would be really interesting. And I've been very inspired by like the transsaccadic priming
with Lisa and Martin. Yeah, exactly. Yeah. That would be cool.
Yeah.
Yeah, thank you so much for the great, wonderful talk. So actually, just jumping off a question just over there. When you're talking about things like navigating the world, and when you talk about like how you're having to constantly kind of engage and disengage your attention from the environments, the instinct that comes to mind for me is that this is like a very kind of cognitively effortful process in a way that is like very rich and naturalistic to investigate, which I find really interesting. So with this in mind, especially when you were just talking now about how this is a process that is engaged over an extended duration, constantly having to refresh one's attention attends to different parts of one's environment, and how this attention, is so interlinked with the discussion that you had of the retinotopy. Would you say that the measures that you are using in your work, like the PRF measures or things like that, can they also be used to index something about whether someone is using like what one could say like an optimal strategy of searching the environment, attending to one's surroundings, and being able to encode this information as cleanly as possible, as heuristically as possible? Or can it also indicate when somebody's acting like in a suboptimal way, like they're just kind of, having to constantly look around, they're really unfamiliar with their environments, it's hard for them, they're getting tired, they want to sit down. So how are you able to get at the cognitive effort that is inherent in this navigational process, maybe using some of the neuroimaging methods you've been discussing?
Yeah, so I think there are a number of really good questions embedded in there. So one of the things that I have seen in the population receptive field modeling literature is work coming out of now I'm gonna blank on his first name, but Clay and John Winnower in at NYU who have been doing some memory-based saccade tasks. So training participants to see an image and know where to fixate based on that image. And then looking at how population receptive field properties kind of can be used and how their responses during the intermediate period where they're recalling that image and where they need to fixate, where how they're responding during that time, and finding that the PRF like representation, especially within the dorsal stream, so in the IPS maps, pretty accurately capitulates what the person's errors are going to be with respect to their fixation point compared to that location. Again, like one of these things that leads me to predict that these are about eye movements and not about something else. So that's IPS. It's not the negative PRFs that I've been talking about. But I'm optimistic about it, that the negative PRFs in particular in the memory areas might be involved because they sit really on the lateral surface. They sit really close to the IPS maps, and that information needs to come from somewhere, so to speak. And I think that this might be a plausible mechanism for reading out that information from memory in order to know where you're going to deploy your eye movements and shaping what happens down IPS. So just like a corollary to all this is something that I've always found, like as PRFs have been used in imagery literature recently, that I've found somewhat counterintuitive to my own imagery experience, which is that in the literature, the couple studies that have used PRFs to index imagery vividness have found that within the visual areas, the activation during imagery tends to be broad, which they have taken to suggest that the visual system isn't getting a high resolution image out of your mental imagery picture. And I think that finding and interpretation is at odds with the experience that people have at least that I have when I do imagery, which is that it is very like high resolution and can be great detail. And I can zoom in and like think about this particular edge of whatever. So we know that we know that high resolution image information is in there. It's just not what the visual system has access to. And what we find when we look at population receptive field sizes, which you might infer as like the blurriness of these images, of the mental image, the negative PRFs in these memory areas tend to be smaller and more foveal than what you see in the kind of higher level perceptual areas, suggesting that they do have a high resolution, image, so to speak, of what is being held in memory that then can be used to can be convolved with the visual system to give the visual system a blurry picture. And I can unpack a little bit more of why it would be important to have a blurry picture in the visual system, but like that's at a high level how I'm thinking about accuracy and optimal versus suboptimal behavior.
Very informative. Thank you.
Yeah. Yeah. Oh, sorry. Oh.
It's being recorded.
You said you also looked at other categories, like objects as well. Did you see anything similar there? And then also, did you look at like different kinds of objects within the object category, like manipulable objects?
Great question. So we haven't done the PRF stuff in object selective cortex, so I can't answer anything about that. What I can answer is for imagery of objects, what we've done or what we've seen. And what we've seen is that there isn't really that much shifting around of object selective activity that's selective in memory compared to what you see during perception. There it looks like there's like kind of activation, but it's like dampened across object cortex more broadly. And that's using object selective, that's using personally familiar objects. So be trying to match as closely as possible what we've done for faces and scenes, but doing it with objects, like objects that you should know deeply and care about a lot. So that I found a little I found is consistent with this idea that scenes are kind of special with respect to imagery. I do think that it's possible that doing a different task on the object, so like imagining an object in motion, then you might get something that's different with respect to how the memory related activity is projected within the motion system, not within the object processing.
Or like
So
if
I
it's an
think there's like
object that
some,
you could use, you know,
yeah,
imagine
yeah,
it's an object.
right. That could also, and then you might expect it to be kind of in this lateral temporal kind of tool use and causal processing kind of system. There you might see some shifting with respect to object imagery. Haven't really done those experiments yet, but I think it's like an empirical question that has like pretty important theoretical consequences for these data. So yeah, great question.
Thank you.
So I noticed in the VR movies you showed us, there was a lot of cue conflict, a lot of distortion of the 3D rendering as they're moving around. And I'm just wondering whether people remember that.
You mean within the photospheres themselves, there's a lot of distortion, or?
So as you moved around,
Yeah.
the 3D representation isn't fabulous. At least not in the movie
Yeah.
you showed us. So there's like a lot of distortion of the 3D perspective.
So it is not.
I'm not criticizing that,
No,
this
no.
was there, and
I'm just making sure. I'm just making sure I know exactly what the. So.
It's really hard in VR.
VR environment is actually just like a photosphere. So it's not something that's like rendered to have depth. It's just like a panorama that they're immersive in. But I think what you're suggesting is that like you're asking whether they remember some of these distortion artifacts.
Yes.
It's
so the straight
possible.
lines were not staying straight.
Yeah.
And I'm wondering whether people remember that.
I wonder if they do. I think it would be interesting.
Because if they don't, this is really important for dealing with motion sickness. I think. Yeah. From VR stuff.
It's a great question. I noticed myself, like when I was working with these images in particular, that if I entered a photosphere and the ground plane was slightly off, or there was like a discontinuity where the panorama wasn't stitched together quite right, that it would make me really sick. So yeah, I think it's a great question. We haven't really looked at it. We did try to choose environments that did not make people sick, or that didn't have these kinds of distortions to have as good of spatial continuity as possible, so that way it wouldn't break this like feeling of being in an environment. But yeah, it's a great question. Yeah, thank you.
Okay, I think we're going to stop now. Thank you. Oh,
there was a question.
Oh, so sorry. Oh. Sure. Yeah, that sounds good. Yeah. Yeah, we'll let you two chat after. But Adam, thank you so much. That was a great talk and great Q&A. And thank you all for being here.
Thanks, everyone. I appreciate it.